All right, I think we're just gonna go ahead and get started. I have about 30 seconds early, so we're gonna start. Welcome to this month's Soil Health Nexus Digital Cafe. I'm Christina Corral. I'm with Michigan State University Extension. I'm one of the directors of the Nexus, along with directors from both Indiana, Wisconsin, and Nebraska. We welcome you, and all of the members of the Nexus. We welcome you to our meeting this month. Um, we're going to start off by just telling you a little bit about who we are, and I'm going to throw up a poll question. I hope it comes up. I do want to just make note that we are a university funded, and so we need our demographics. I'm going to put that up as we go along. We are the Soil Health Nexus is a university-led group. We are in the North Central region. We do have other members who are Nexus who are not from the university, but we do welcome anyone. We're open to anybody who wants to be part of us, and we are again from the 12 North Central regions. So our digital cafes, for many of you, you already know this, but for newcomers, welcome. We are setting up as a presentation. We'll go through today's presentations and we'll have breakout rooms for further discussions on what you've heard today, and it's very open. And those breakout rooms are researchers, education, agency staff, farmers, and students. Okay. So today I'm going to stop the poll in about 50 seconds. So those of you who haven't done it, please hurry up and get them done. Today, Brian Dougherty from Understanding Ag, and that name is probably familiar to a lot of us. Brian, when he was with the university in Iowa, he was actually one of our directors for the Soil Health Nexus, and we're excited to bring him back. And he's going to talk to you about biological approaches to nutrient management. And with that, Brian, I'm going to stop my sharing if you want to start your sharing. I think you are ready to roll. It's a blank background right now, Brian.
Okay. Let me try that again.
All right. Perfect.
Okay, how's that looking?
It looks really good.
Okay. Yeah, so as Christina said, I'm Brian Dougherty with Understanding Ag Consulting Company. And kind of a big topic today, biological approaches to nutrient management, and also through water quality in there, because I know that's a big interest to a lot of people in the group here. So for the sake of time, I'm going to do my best to get through this in 30 to 45 minutes. Hopefully we'll have a little bit of time for some breakouts, but I'm just going to jump right into it. So what I want to get into today is just talk about some of the challenges I've seen just in the last year and a half traveling all over the country, nutrient management, water quality, a little bit on how do we fix it, and really want to get into more of a biological approach to managing nutrients and how we can leverage that. So another poll for you. So sharpen your pencils here. Why do we have water quality problems? So is it A, we haven't thrown enough money at it? B, we just need to put the engineers in charge. Now, I was trained as an ag engineer, so I want to pick this one, but unfortunately, I don't think that's going to get us where we need to be. I think we all know it's not just the golf courses and geese that are the problem. D, we hear this a lot. We just have leaky soil and it's just really hard to fix. Not much we can do about it. Or what I'm going to pick as the answer is actually E. It's because our soil is dysfunctional. And just as a broader agricultural community, it's just really difficult to fix this at scale for a whole bunch of complicated reasons. Now, I don't have, a magic wand. I can't fix all of these complications, but I do want to talk about soil dysfunction and how we fix it and maybe ways to get over some of these hurdles so we can make progress at scale. So if dysfunctional soil is a problem, then what do I mean by functioning soil? So I've started using the term soil function more and more instead of saying soil health, because soil health, it's kind of a nebulous thing and a lot of people don't know what you're talking about. What we really mean is what is that soil doing and what is it doing for us? So number one, it's got to be well aggregated. We need rapid gas exchange. We need fast water infiltration. If your soil is not well aggregated, it's pretty much dysfunctional. We have to be able to get water into the soil and fast gas exchange, minimal compaction, good microbial activity, rapid nutrient cycling, and lastly, produces healthy disease and insect resistant plants with no side effects. Now, a lot of people think that that's kind of crazy and we can't do this without fungicides and pesticides. That's a topic for a whole different webinar. I'd love to get into that. Don't have time today, but I do know producers that are doing this without pesticides, without fungicides, without giving up yield. So it is possible. We can get there. Some of the challenges I see pretty much everywhere I've gone, East Coast to West Coast, Midwest, doesn't really matter. Number one, compaction. See it everywhere. Soil just can't breathe. Number two, broken water cycle. I'm going to talk about what I mean by that. It's just not functioning. Number three, we have an over reliance on pesticides. Our soil is sick, our plants are sick. And so we're using these, techniques or tools to try to address that problem. Number four, a lot of nutrient management issues. Our soils are constipated, or we say they have diarrhea if we have nitrate leaching. And so then we step in with nutrient management and try to overcome that essentially. And number five, lack of carbon. The biology in the soil is essentially starving to death, doesn't have enough carbon to eat in a lot of these systems, which leads to number six, lack of biological activity. And if the biology is not functioning, your soil just cannot function properly. But when I put this list together, I realize, just thinking about the interconnectedness here, number one and two are really just symptoms. Soil is not inherently compacted or lacking in aggregation. It got that way usually because of our management. So then number three and four are really just reactions to the problem. That's what we do as a management technique to step in and try to fix it. But it's really number five and number six that are the fundamental problem in almost all cropping systems that I've seen. Lack of carbon, lack of biological activity, and unless and until we deal with number five and number six, we're always going to be fighting the battle with those first four. So that's what I want to talk about. So what do I mean by a functioning water cycle? Well, you can see it here on the right, basically green living ground cover, residue cover, soil armor. We've got good infiltration. We've got good aggregation, and we need our water transpiring through plants rather than just evaporating off of bare soil like you see on the left, where you've got compaction, you have a lot of surface runoff. The left side is a completely broken water cycle. The right side is what I mean by a functioning water cycle. So bare soil, I'm sure you're all aware of this. It's quite devastating to soil life and really all life if we think about it. So this is just a fun little picture I snapped at a field day up in Wazeeka, Minnesota with a thermal imaging camera. You can see a soybean field here. This is about 10 o'clock in the morning, low 80s air temperature. That bare soil is already 103 degrees. The surface of that soybean plant was 69. Now, that is the power of plant transpiration. That's why we need our water going through our plants and not evaporating off of bare soil, because it has that cooling effect. And a critical point here is that right about 105, 110 degrees, somewhere in there, we actually start to break down enzymes and proteins. We're literally starting to cook the biology out of the soil. And we regularly see surface temperatures 130, 140 degrees in crop fields. Then we have the pesticide issue. So I think of pesticides as kind of a circular firing squad. And I don't know, a lot of people are probably going to disagree with this, but we know we need a thriving, diverse biological community in the soil in order to have good plant function. So a disease or some insect pressure shows up. So, we apply a product essentially to fix the problem. It goes away for a while, but the challenge is, those products don't just target that one organism. If we could develop products that could do that, that would be a huge win. But we know they take out other things, which suppresses the biological community. We know plants need a healthy biological community. So now we've got plant health issues, which causes more disease and insect pressure to show up. And then we fix it and around we go. So it's really kind of an ass backwards way to solve a problem when you think about it. I mean, we've got to figure out how to get off this treadmill. And that's the topic of plant health that we can hopefully get into another time. But this is doable. It's not easy and it takes time. But we do have producers that have been able to get off this treadmill. So lack of carbon, you're all, I'm sure, very aware of all of this if you're part of the soil health nexus, but most of our stable organic matter is coming from root exudates and fungi and dead microbe goo and pretty much everything that's, available for microbes to eat as a carbon source. One way or another, it's coming from plants or it's coming from other microbes. This is all biologically driven. So we just need basically more plant growth out there. Lack of biological activity. We see a whole lot of this around the Midwest in the fall. So just think about how many months out of the year do you actually have a living root in the soil? Maybe 4 or 5 with corn and soybeans where you could actually consider that root to be alive. But more importantly, how many months do we actually have active root exudation where those plants are pumping carbon into the soil to feed that biology? I've heard people say in corn, it's even as little as 6 weeks. It's not as long as we think. 6 to 8 weeks maybe is where we've got really active root exudation happening. So the question is, what's happening the rest of the year? What is our biology living on? It needs carbon as its food source. So if we don't have any root exudates, and we get the situation like you see in the middle here with no ground cover, there's not a whole lot of groceries out there. So that biology is going to start eating our organic matter, or it's going to eat the glues that hold our soil together, or it's going to go dormant because it doesn't have anything to eat. So the question is, how do we fix it? So these are all pictures that I've taken in the last year and a half, just in different fields I've been in all over the country. So we've obviously got some serious degradation out there. Luckily here in the upper Midwest, things look a little bit better than this, but this is a pretty widespread issue. So I start with the premise that dysfunctional soil and poor water quality, they're fundamentally biological problems and they require a biological solution. That is the only way we can fix it. We're not going to buy our way to healthy soil and clean water. We're not going to fertilicide our way to it. We're not going to engineer our way to it. The only pathway where we're going to fix soil and water quality at scale is to grow it. We need green, living ground cover throughout the growing season over most of the landscape if we're going to get there. So pathways to regeneration. This is just kind of a different way to think about it. This is from Professor Stuart Hill. He came up with this ESR model, he calls it. So number one is efficiency. So in an agricultural context, here we're basically talking about, the 4 Rs, inhibitors, reduced tillage, things like that, all good practices, but they're really just treating symptoms. They're not addressing the root causes of dysfunctional soil. They can be a good step in the right direction, I would say. But if you stop there, it's basically a barrier. It's not really allowing you to make progress. The second step is substitution. Now we work with a lot of growers that are doing this sort of thing, humic products to stabilize nitrogen, different forms of nitrogen, making biofertilizers, a much more progressive step. This is all fantastic. I highly encourage it, but we can't stop there. That can still be a barrier if you just get to that point and then don't do the third step, which is actually redesign. And here we're talking about the soil health principles. I just call it the 6 Ps for the 6 soil health principles. Instead of the 4 Rs, we need the 6 Ps basically because they address the root cause of dysfunctional soil. Now, you all know what the soil health principles are. I'm not going to spend time on that. But the reason they work is because they build aggregates. They add carbon and they feed biology, which, going back to my earlier slide, those first 4 problems, the soil health principles take care of those. They fix our compaction, our broken water cycle, they cycle nutrients, and they help us grow healthier plants. So we've really got to implement the principles across the landscape. Not easy to do, but we can get there. So aggregation, you're probably all aware of that, but I think of aggregates as like the engine that drives soil function. It's just absolutely fundamentally critical for soil health and soil function. We have to have aggregation because that provides all the goodies we need. We got water for the biology to live in, aerobic, anaerobic pockets. We can have nitrogen fixation happening right next to where a root is. High and low pH, high and low redox potential. That's a whole new frontier that we're just starting to get into. Looking at redox, it's every bit as important as pH. It's just hard to measure, so we haven't really gone down that road. but we've got to have aggregation. So we kind of had this old model of soil function. When I first started taking soil classes in the early 90s, this is kind of how we thought about soil. We knew we had our chemical, our physical, sand, silt, clay, NPK. The soil was just kind of there to hold up the plant. And then we knew there was biology out there, but biology back then was mostly thought of as a detriment. Basically, if it moves, kill it, right? We don't want these creepy crawlies eating our crops. We didn't really think about them as being an important component of the system. So now we think about soil like this: chemical, physical, biological, they all overlap. I'm sure you've all seen this graph. And where they overlap is this thing we call soil health. But I want to challenge everybody to think about soil a little bit differently. If this is the way you think about soil health, I want you to erase that from your brain. I know we've been talking about this for years and years, but I think this is how we need to think about soil health. It is fundamentally a biological system. Biology dominates in a healthy soil. Think about what is hands down the most important physical characteristic of soil. It's how well aggregated it is and what builds aggregates. Biology, plants working with microbes. These chemical reactions aren't just happening in a test tube. These are biochemical reactions. In a healthy soil, again, a good majority of your nutrient uptake by plants is mediated by and goes through the biology, and we've got to have carbon to make the whole thing work. So when I think about soil health, I think about carbon and biology. If biology is not the dominant driver by far, then your soil is dysfunctional. This is how I think about soil health. So I would just challenge everybody to kind of think about soil differently. So getting into nutrients. We don't often think about carbon as a nutrient, but I call this the most important cycle on your farm. This is just a busy graph. I get it. This is my engineer coming out. We like our boxes and arrows, but this is just a carbon cycle for a 200 bushel corn crop. The numbers don't matter. I'm not going to go through all this. The point I want to make here is, we've got carbon coming in through the crop. It's all just cycling through the system. But where's the fertilizer truck? There really isn't one. Unless you're adding a lot of compost or a lot of manure, we add very little carbon back to our soils. And that's fine. We don't have to add carbon if we're growing it. What we do need is generally just more plants, more green living cover. We can have a new arrow going into our box. And now if we want to get into the carbon sequestration, talk or just increasing organic matter, we've got to have new arrows going into the box here if you want to make that box bigger. But the point I'm trying to drive home here is this is all biology. Biology is driving the most critical processes on our farms and in our soils. So if we look at the field scale, when I'm talking to producers, I really hone in on this because, we hear a lot about climate change, extreme weather. They may or may not care about this. What they need to care about is what's the carbon cycle in their field and how does it work? So we all know, maybe there's some hort people in the crowd here. I haven't figured out how to grow a plant with a potato below ground and a tomato above ground on the same plant. Maybe you've gotten there, but it's a nice graphic that illustrates the point. So we've got photosynthesis. Plants using CO2, it's making sugars. It's pumping some of those sugars into the soil. The microbes are eating it. They're consuming the carbon. They're consuming the oxygen, and they're releasing CO2 from the soil. And this is, I think, the critical piece that a lot of people don't really think about or overlook is that plant is literally feeding itself. It's a mirror image of itself. The exact opposite is happening below ground that's happening above ground. So in an efficient system with healthy soil, we want our plants getting their CO2 from the soil, not trying to hoover it out of the atmosphere. And this, just as an aside, we run a lot of Haney tests, and I know they're very controversial. A lot of people don't like them, but this is why we run Haney tests. I want to know what that number is. Is that soil breathing or not? If your CO2 respiration's low and that soil can't breathe, it's like having a plastic bag over your head with a couple of holes poked in it. You're not going to be a very happy camper. The soil's not happy if it's not breathing. So what happens when we've got compaction and poor aggregation? the whole thing kind of goes to heck in a handbasket. The roots can't breathe. The water cycle doesn't work. Plants aren't happy. Microbes aren't happy. I think of aggregation as a survival mechanism. Plants are actively trying to build aggregates because that's the environment they need to survive and thrive. So our job as land managers is basically to get out of the way and let plants build aggregates. So water, calling this the second most important. You can argue carbon or water is more important, doesn't really matter. But again, just simple water cycle schematic here, 30-inch rainfall environment, doesn't matter. The point here is what's driving this? Evapotranspiration, a vast majority of that should be transpiration, not evaporation. That's driven by biology. What determines how much surface runoff you have? Well, really, it's how well aggregated is your soil and how much water can it hold, which again is determined by aggregation and biology. What determines how much water your soil can hold? Yes, there are definite textural differences, sand, silt, clay, but we can't fix that. What we can do is increase organic matter and aggregation and pore space and increase our water holding capacity. So again, I'm going to keep harping on aggregation. It is just absolutely critical for a functional water cycle. Bit of a trick question. What drives precipitation? Nothing we can do about that one, right? Well, actually, I would argue that over land areas, it's actually biology that's driving this. This is the small water cycle we talk about. It's actually microbes and volatile organic compounds in the atmosphere that are driving a lot of the raindrop formation. They're the nuclei for raindrop formation. The numbers are vary depending on the research you look at, maybe 50 to 80% over land areas. And so when we denude a landscape, we take away the vegetation, bare soil for months at a time, we're essentially breaking that biotic pump. And that then creates wet and dry extremes. We kind of create our own problems in a way. Now, if you got a quarter section, you're not going to change your rainfall patterns. That's just never going to happen. But we can do this. You get to the scale of, hundreds of thousands to millions of acres, which, we got what, 25 million acres of corn and soy just in Iowa. We could start changing this and you could start seeing a moderation of some of these wet dry extremes. So getting into the biological nutrient management, I just want to hammer home it's the management that really has to be in place to make this work. This is a very complex system. You're managing a lot of moving parts and it's continually evolving and changing. And that's a lot of people just don't want to deal with that. I get it, but we need to get there, as an agricultural community. 3 points here. Number one, follow the soil health principles. Number two, address factors that limit productivity. Number three, we've got to maximize photosynthesis. Now, these all go hand in hand. No one's more or less important than the other. They all feed into each other. We can do them all at the same time. And now this is going to make a lot of people very uncomfortable. And you're probably going to want to reach through the screen and choke me for saying this, but we really there's no prescriptions for this. The recipes that we've been using for decades, you pull a standard soil test, you look up some numbers in a chart for removal rates. That's really an antiquated way of thinking about plant nutrition. I'm going to talk about why, but we really need to kind of scrap that and just completely rethink how we're managing nutrients in these biological systems, because our fertility decisions, we have to at the very least account for biological function, which standard soil testing doesn't do. But ideally, your fertility should be benefiting biological function. A lot of our fertility in what we would call a conventional system is actually inhibiting soil biology. So number one, soil health principles. If you boil it down, this is all about just nurturing the soil food web. We can start weaning off of some of these inputs that are inhibiting biology. And I really believe that we have to eliminate, not everywhere all at once. It's going to take time. Not saying we can do this overnight, but we have a number of producers that have completely eliminated fungicides, insecticides, and seed treatments, and they're not giving up yield. This is doable. Just takes a little time to get there. We can back off herbicide rates. And I can talk a little bit about how to do that, reduce purchase fertility. All these things are doable. We've got to keep the biology alive and then use that biology to address our limiting factors. I would say hands down, pretty much everywhere I go, the number one productivity limiting factor on most farms is air exchange. The soil cannot breathe. We've got to fix the compaction and build aggregates using biology. We can't do it with steel and tillage. Number two would be water, and air and water are just flip sides of the same coin, again, because they come back to aggregation. Number three is probably decomposition. The residue's not breaking down and the nutrients aren't cycling. I see that everywhere. And then number four, there could be some nutrient that's actually limiting productivity. That's often maybe a micronutrient, or it's actually a nutrient antagonism. We put way too much of something on and it's inhibiting the uptake of something else. But, as an industry, we tend to, try to jump right to number four. We're just going to put a nutrient out there and we're going to fix it. We've got to pay attention to those first three before we try fixing it with a nutrient. It's really the soil microbiome that limits plant productivity. It's all about the biology. Number three, maximizing photosynthesis. So the question there is, what limits photosynthesis? I talked about this earlier. Look at the equation. What do you need? You need CO2 and water. So what is likely to be limiting your photosynthesis? It's probably either CO2 or water. How do we get more CO2? We increase microbial respiration from the soil. How do we get more water? We build aggregates. And then number three, energy capture. There's all kinds of ways through cover crops and relay cropping, multi-cropping, perennial ground cover systems, adaptive grazing. There's a whole laundry list of ways that we can basically just grow a bigger solar panel and have that solar panel out there throughout the entire growing season so we can ramp up that photosynthesis. And then again, number four, there could be some nutrient that's, limiting photosynthesis. Manganese is a classic example. You have to have that to split water. But that little arrow there represents a whole bunch of complicated stuff that happens in the plant. And again, we can look at the micronutrients. We need to not just focus on NPK. There could be some antagonisms or deficiencies that are limiting us there. And we can, identify those through some, tissue testing or plant sap analysis or things like that. So there's kind of this old view out there of how plants acquire nutrients. And this is all still true, all still happening. But this used to be kind of the whole picture, or at least this is how I was taught, 20, 30 years ago. You've got passive and active transport of nutrients. Basically, plants are taking up the nutrients through the soil water. And they can pump hydrogen out, change the pH in the root rhizosphere, and make some things soluble. It was typically just kind of taken as for granted that plants don't really use organic nitrogen. We know that's not true now. It's really kind of a hydroponic system. The soil is just kind of there to hold the plant up. And I don't know who originally said this quote, but I think it really kind of hits the nail on the head. If you farm like your soil is dead, you're going to create dead soil. We've got to farm in a way to keep our biology alive and healthy. So in a biologically inactive soil, this is pretty much the name of the game. This is how you're going to get nutrients into plants. But what if you have biologically active soil? Just as a catch-all term, I'm just going to call this microbe farming. Plants are actively cultivating specific microbes to acquire specific nutrients when they need them through changing the root exudation. A good majority, we don't have an exact number, depends on the nutrient, depends on how healthy the plant is and soil, but easily 80+% of plant health and plant nutrition is driven by biological functions. It's actually microbes, fungi, things like that are getting the nutrients into our plants. We know now plants can readily take up organic nitrogen. And I think this is absolutely key. If we're going to deal with our water quality issues, we've got to get to a cropping system that primarily relies on organic nitrogen. Now, this passive and active transport, it's still happening, but it's just a smaller piece of the picture. So here, you farm like your soil is alive, you create living soil. This is a self-reinforcing system. It gets better over time. So I usually ask for a show of hands here. How many people have heard of the rhizophagy cycle? If you have not heard of the rhizophagy cycle, you have to Google this. Dr. James White, rhizophagy, just look it up and start studying this, because I think this is going to absolutely revolutionize how we manage plant nutrition. Plants are literally farming and eating microbes. They're carnivores. They're actually, through that root exudation, they're pumping carbon out the root tip. They're actually ingesting bacteria and stripping off the cell wall and using it for plant nutrition. And what's even crazier is those little root hair formation that you see here, that's actually triggered by the microbes inside the root. So the plant's spitting some of the nuclei back out that actually survive this process with a little bit of root exudate, and they reform their cell wall and go on their merry way and possibly be ingested again. This is the cycle. And this is how nature works. This is how plants get nutrients. You know, in cropping systems, the way we manage, through genetic selection and high input, high fertility type systems, we've kind of bred a lot of this out of plants, their ability to do this, their ability to associate with mycorrhizal fungi. We need to start thinking about plant genetics and plant breeding differently so we can get this to work. And more importantly, why I'm so down on seed treatments, guess what inhibits rhizophagy? Seed treatments. Antimicrobials on the seed, chelating herbicides, high salt fertilizers, all the things, excessive nutrients in the root zone, all the things that shut down soil biology and the plant's ability to do this. Essentially, again, we need to kind of get out of the plant's way and let plants be plants and let biology be biology and do their thing. So there's this paper that came out a couple of years ago talking about how nitrogen is it's an emergent property of healthy soil. We see this all the time. Don't have time to go through this, but there's a whole laundry list of ways that plants can access nitrogen that have nothing to do with the fertilizer truck. Plants can do this if we create the conditions in which they can do it. So, historically, we thought, free living nitrogen fixers, maybe 5-10 pounds, per acre. So we just kind of ignored it. It was a rounding error. I would argue that we can do a whole lot better than that through all of these different processes. But I don't think we even really know what the potential is out there. But we think about nitrogen, we have this large pool of organic nitrogen in the soil that we can tap into. So you can measure this with a total nutrient digestion test, but say I'm going to grow corn, we come in, I put on 200 pounds of nitrogen, the corn takes up about 200 pounds, about 80 goes back in the residue. So I put on 200, the corn only used 120. Where'd that other 80 pounds of nitrogen go? Well, it probably either leached or denitrified or volatilized, or what we want to happen is that nitrogen stays in the organic nitrogen pool. We convert it to organic nitrogen and make this pool bigger. So I think of, fertilizing, it's kind of like peeing in a swimming pool. It feels good, but it doesn't really add much to the pool. And the public would be happier if we'd all just quit doing it, right? So that was a joke in case anybody out there's not awake. It's hard to tell jokes over Zoom. But so then we grow a cover crop. Just say we throw some rye out there. It's not a miracle worker. It's not a legume. It can't, create nitrogen, but it can stabilize nitrogen. It can take this leaching loss we have here. It basically helps plug the leaks. But really, if we want to make this pool bigger, we need more carbon to go with this nitrogen. We need diverse cover crops. We need those free living nitrogen fixers. We need that rhizophagy cycle working. So if we look at nitrogen and nitrogen uptake by plants, there are more and less efficient ways to get nitrogen. Hands down, the most efficient, it's basically 100% efficient, is to let that plant eat microbes, because we don't have to do anything in order for that to happen other than get out of the way. Nitrate is the least efficient form of nitrogen, but our cropping systems, we're basically swimming in it because we put a lot of readily available high doses of nitrogen and ammonium in the soil. And then if you don't have a living root there to use that right away, it can leach on us. So that's, one of the big challenges. We can shift this and use more, organic nitrogen sources and keep that ground cover out there. We can deal with a lot of these nitrate leaching issues. We got to focus on cycling our nitrogen, growing it with legumes, with the soil biology. And that can enable us to use lower nitrogen rates. We need to focus on nitrogen use efficiency. You've all probably seen these numbers, book values somewhere around 1.2 pounds of nitrogen. Most producers we, first start working with, they're using about a pound of nitrogen per bushel. If you use the nitrogen rate calculator tool, depending on prices and, yield that you put in, probably somewhere around 0.8 pounds of nitrogen per bushel. But we have a lot of producers in the 0.4 to 0.7 range. So this is entirely doable. People are already doing this. And the way they're doing it is basically just healthy soil. They're building aggregates and they're just increasing that soil's natural ability to produce nitrogen. And you don't have to give up any yield to do this. It just takes some additional time and some additional management. So Dr. Franzlever's research, he was probably already showed this on a Nexus webinar. I know he's been on in the past, but just want to reiterate, we already have the data that basically backs this up. Soil respiration, just look at the graph on the right here. The more biologically active that soil is. Now, this is a 3-day test, so these numbers are going to be higher than what we see on the Haney test, which is a 1-day. But the more biologically active it is, here's nitrogen use efficiency, pounds of nitrogen per bushel, the less nitrogen we need. It's right there in the data. So I don't have time to go through all the other nutrients, phos, potassium, but just in general, we need to, really put a lot of emphasis on what we're putting in the soil. We've known about salt indexes for decades, but unfortunately, the cheapest fertilizers tend to be the ones that have the highest salt index. So we need to kind of rethink, potash, potassium chloride, for example, widely used because it's the cheapest, but it's not the greatest thing for soil. There are better and worse forms of these different nutrients that we can apply. This is not a perfect list, just kind of ballpark. Even our livestock manures, there's a lot we can do stabilizing those with more carbon, with some biological amendments to treat that manure and make it more biologically friendly. We got to pay attention to application rates and all that, but we really need to put more emphasis on biology friendly plant nutrition and also think about adding some carbon. We really need to be stabilizing, especially our nitrogen. Any kind of liquid nitrogen you're putting out really needs to be stabilized, ideally with some humic acid. That's a carbon product, a little bit more natural, more soil friendly. All of our foliars could have fulvic acid in them. If you're not familiar with these products, you can do the research. There's some snake oil out there. You got to be careful who you're buying it from. A lot of people are making their own compost now. They naturally contain a lot of these fulvic and humic products, but generally they're really good for our soil. And we can use these to reduce herbicide rates. And a lot of people are nervous about that too, but it's doable. And it's the point here is we're not just going to completely abandon inputs. We're just going to try to get the soil biology to replace their function. And especially, again, I think the seed treatments are critical. We could be replacing those with biologicals. Don't do this year one. You got to get your soil working, but you can get there over time. We got to think about feeding our soil livestock. Everybody has livestock, even if you can't see it walking around. So this thing we call farming here on the right side, unfortunately, our pesticides, high salt fertilizers, tillage, poor grazing, compaction, all of these things inhibit our soil biology. So our job as land managers is just figure out how do we do less of this and more of this stuff on the left side. This is not a complete list by any means, but we know how to feed soil biology. We know what's friendly. We know what keeps it alive and going. We just need to be doing more of that because, every acre essentially, you've got probably somewhere between a half a cow and 2 cows per acre that you have to feed before your crop gets to eat anything. In a biological system, we have to feed the biology first and then let the biology feed the crop. That's where we're trying to get here. So then we're primarily relying on organic nitrogen or other organic forms, and it's just going to be a better system. So I'm going to lay out the argument here that functional soil is the only scalable solution for water quality. We have to implement the principles across the landscape with a priority on maximizing living roots and photosynthesis. We have to think about nutrient management as an integrated system. We have to keep the biology alive. We've got to stop killing it. I talked about these other things, organic forms of nitrogen, small doses, when the plant needs it. We can do a lot with foliar nutrition, lower salt indexes, stabilizing our nitrogen, stabilizing our manures. And then again, number 5, I touched on that earlier. We can absolutely grow plants without fungicides, without pesticides. We're not going to get there overnight, but it's doable. We just can't do it with the same management that we've had in the past. That will be an absolute train wreck. You have to change your system if you're going to take those products away. So just throw out a little challenge. Let's prove or disprove it. Make me eat my hat. whatever. I'll come back in 5 years if I'm wrong. But I would love to have, a research site or multiple sites where we compare these different systems and look at a systems approach. We can't just, put in a cover crop or change nutrient timing and see what happens. I get it. That's how we do research. That's how we get published. But we really need to just a completely different system. Everything's going to be different. And let's just compare them head to head. You're going to have probably different equipment, even different timing, different forms of nitrogen, different application rates. All of that's going to have to change. But I would wager that within 5 years, pretty much any metric you want to pick, I don't care what it is, environmental outcomes, soil metrics, profit, that regenerative system will outperform the conventional one. Now, it takes time to get there. That's why I'm saying 5 years. Certainly not going to do it right away. The only one you may not outperform would be yield. And, personally, I don't really care about yield that much. I care about profit. The last thing we need is to grow more corn, more soybeans, overproduce even more, drive the price even lower, and create even more economic stress for our growers. So I would just challenge everyone. We can absolutely do this. I think we can do it in a way that benefits pretty much everyone, even the fertilizer and chemical companies. There's things they could do to change their business model and tap into this. And I think we can do it at scale. And this is incredibly ambitious, but I think we could meet our nutrient reduction strategies within a decade if we get serious about this. But it's going to require every one of us to put 100% of our time, energy, and resources into fixing dysfunctional soil, because that is the fundamental problem. Everything else is just kind of a distraction. So I know I went through that really quick. I hope we got a few minutes left, but just resources. If you want to do a deeper dive, I did a 5-part blog series on this. You can go to our website, understandingag.com, and just look for our blogs. I've done a bunch of them on there talking about carbon cycling, erosion, how do we deal with compaction, making the transition, all that's up on the website. So with that, hopefully we got a few minutes left here. There's my contact information. Happy to do questions or go into breakout rooms. So I appreciate everybody tuning in today, and hopefully you took something away from this. Reach out with comments, criticisms anytime.
All right. Thank you, Brian. We do have a couple of questions. So I'm going to start with those before we go into breakouts. Scott Satterwhite is asking, how do you think labs could reduce their costs for PLFA tests? Would it be take new technology or more people?
Oh, that's a good one. You'd have to ask a lab person that. I don't, I know they're expensive. They're about $80, unfortunately, but yeah, I suppose just more efficient processes. Yeah. Call Lance at Regen Labs and ask him. Sorry, I don't have a great answer.
We're going to continue on with Scott. He's got another question for you. Do you think producers could better understand the biology need if they started with adding actual biology? Then they could move on to the 6 Ps. Additionally, do you have any thoughts of adding actual biologicals versus products that encourage increased biological activities?
Yeah, that's definitely a strategy. We have a lot of growers doing that. Again, they're putting biologicals on the seed. They're putting stuff in furrow, compost extracts. I'm a big fan of all of that, but I would just say we need both at the same time. You've got to have the living root out there to keep carbon pumping into the soil to keep that biology alive. Otherwise, odds are something's going to kill it. It's not going to survive if it doesn't have carbon to eat. So yeah, I'm in favor of that technique, but just that alone, I don't think is going to get us where we need to be.
All right. Marcel would like to know, can you talk more about the chemical companies could do to benefit? Oh, sorry. Could you tell more about the chemical companies could do to benefit from a transition to regenerative ag? Hard time speaking.
Yeah, that's a tough one. At more of the local regional level, the co-ops, I think there's just a huge potential there for them to get into the cover crop business. They could be, harvesting seed, helping people with that whole process, cleaning seed, making more seed available. So the big business opportunity there that could potentially offset, I'm not saying we're going to cut out fertilizers. You're still going to use things, but we can just use different forms. They could carry different products that are more biologically friendly and make those more available and get into the cover crop side of it. On the chemical side, a lot of those chemical companies are also seed companies. So the way I see the potential for them to benefit is, developing new genetics that thrive in these more low input systems. We really need corn and bean hybrids varieties that do well and make good, strong connections with mycorrhizal fungi. That's something that could be measured, and they could put that right in their seed catalog. They have all these things for Goss's wilt and whatever. They've all got rankings on them. I would love to see a ranking in there for how good that plant is at connecting with the soil microbiome. So that's something that those larger multinational companies, I would love to see them get behind that. I think there's huge market there.
Nathan is asking, what do you see as the first small steps for traditional row cropping to transition to the regenerative practices you discussed?
Yeah, first small step would just be a cover crop to plug that hole in the growing calendar where you've got bare soil. So, after in corn beans, I think it's almost a no-brainer to just grow some cereal rye before you plant beans. Put it in after corn. It's pretty much bulletproof. It'll almost always grow, even if you don't get any growth in the fall at all. And beans are very tolerant of cereal rye, so that would be by far the simplest place to start. And then corn, then it's a little trickier, trying to get a little bit more diversity ahead of corn, maybe looking at some shorter season corn hybrids so you got a little bit more time to get that cover crop in. But that's where I'd start, just getting the ground covered. That's our fundamental problem. We just don't have enough ground cover. We don't have living plants out there.
Thank you. The last question, well, I guess we have two more, but we're gonna do one more, and then the other one, Christine, you can ask Brian. It's on biochar. What about using biochar to add carbon food to the soil, especially on degraded soils?
Yeah, I'm a fan of biochar. Nothing wrong with it. It's a tool. We can use it. It works, I'd say, quite well in degraded soil, but I would just encourage anyone, if you're going to use biochar, inoculate it with some biology or nutrients or both before you put it in the soil, because it can act as a pretty strong sponge and you can take a yield hit initially. But that's a tool. I think there's a lot of forestry waste and things that we could be making biochar out of and putting it on our crop fields. But in the long run, we need to see more biology out there. Biochar is one of the ways we can create more of a home in the soil for that biology to live in. So I'm not against it. The big challenge is just expensive. So we got to figure out maybe ways to get it right in furrow. That's where I'd start. If you can do that, just a little bit in furrow instead of doing a broadcast application, I think we'd see a little bit better return for less cost.